Hi, welcome and thank you for joining part 4 of the Techniques series on multi-laser nanoparticle tracking analysis, NTA. My name is Julie Chen Nguyen, and I'm going to facilitate today. So Dr. Jeff Bodicombe, our product line manager for particle characterization, will be our substitute speaker today. Unfortunately, Dr. Travers is not able to be here. If you haven't seen our part 3 of the series on DOS, where Jeff has presented, I would encourage you to hop over to our website for the recorded version. Okay, we'll have a Q&A session at the end of the webinar where you can ask Jeff any questions that you may have. All right, so I think we're good to go. Jeff, whenever you're ready.
Great. Well, as Julie mentioned, this is Jeff Bodicombe. I'm filling in for Dr. Sean Travers in his talk on multi-laser nanoparticle tracking analysis. And so he put together a really great talk, so I'm filling in for him is pretty easy, and I'm happy to do it. He might be in the chat box later on for the Q&A session, kind of depending on things. So let's get to it. So quick outline of what we're going to do today is we're going to talk briefly about what is nanoparticle tracking analysis and why you want to use multiple lasers. Talk about what makes the ViewSizer unique, and then we're going to move into how to profit from that. So biotech applications with viruses, production in gene therapy for infectious titer, quality control, stability testing. Moving on to extracellular vesicles and exosomes. Kind of closing with some liposomal adjuvants, which seems to be vaccine work seems to be top of everybody's mind these days. And then we'll do a wrap up with conclusions and then a Q&A session. Kind of start with what problem are we solving? If you look at nanoparticle characterization, you'll see some unmet needs. The first is visualization, particularly of polydispersed nanoparticle systems. And then second, accurate reproducible measurement of number, concentration, size, distribution details, and some kinetic processes. In this talk, we're really going to focus on the first two, the number, concentration, and size distribution. A problem we have is that with nanoparticle systems, you can't image the particles directly, and so you're going to use some sort of scattering phenomena. And whether it's dynamic light scattering or laser diffraction, anyway, it's going to be scattering as you go below a couple of microns. When you're looking at a range of particle sizes, you're going to see that the scattering intensity is a very strong function of particle size. So angular scattering coefficient on the left as a function of diameter, I can see an 8 order of magnitude change in scattered light intensity with a 2 order of magnitude change in particle diameter. This really prevents accurate tracking, sizing, analysis of samples with varying particle sizes, and this problem becomes more difficult as your sizes get smaller. Unfortunately, that happens in a fair number of samples of interest, and especially in biotechnology world. So this is a problem that really kind of affects a lot of work in biotech. It affects other areas, including synthetic nanoparticles and so on, but most of today's discussion is with the biological applications. It's a well-known issue. If you look at ISO 19430, you can see that they write, polydispersity affects the ability to track and therefore analyze different size fractions. And so in a polydispersed sample, large particles scatter a lot more, making it difficult to detect or track small size particles. So this is the ISO standard for particle size analysis. ISO calls it particle tracking analysis, but many people call it nanoparticle tracking analysis to clarify that it's most commonly used with nanoparticles, but doesn't have to be. Quick comment from one of the early customers, Dr. Eisner at OHSU in early cancer detection research, that it's the first product they found that really can effectively characterize particles in polydispersed samples with a range of particle sizes. And so he's pretty happy, and we have a number of other people that are pretty happy with us, so we're happy. Good to know the technique is being accepted. How did we solve this issue of strong scattering as a function of particle sizes? Well, it really broke the problem up into manageable segments. So we look at the smallest particles, the middle-sized particles, and the big-sized ones. And then we have to look at everything at once and glue everything back together. So looking at everything at once, what we did is we had 3 different color lasers with different intensities. A red low-power laser, which works nicely for the largest particles, a blue high power laser, which does well for the small particles, and then a green laser that gets the particles kind of in the middle of that size range. So we'll call this multispectral advanced nanoparticle tracking analysis, MANTA. Whenever I say that, everybody thinks of ocean wildlife, and so I just call it nanoparticle tracking analysis, or NTA. But it is a distinct technique in the sense it uses 3 simultaneous lasers. The internal layout is shown here. On the left side, you see 3 different lasers, red, green, and blue. They bounce off a mirror through a slit, some focusing optics into the sample, which is a square cuvette. You have the particles, and then moving along the right side towards the back, you have another focusing objective, and eventually a detector on the video camera, which tracks the scattering from the particles as they move about. A nice little package. There's what it looks like. It's your basic instrument box. You're not sitting here with an optical bench or any silliness like that. You open the door, you put in the sample, and away you go. Now, in terms of sample presentation, putting it in, first we measure 25 short videos, and the reason we do that is we want to measure a lot of different particles from the same sample in order to get good statistics. So if we took one long video and looked at the same particles over and over again, it really doesn't tell us as much about the size distribution and compared to looking at multiple sets of particles. So you use a standard cuvette with a black insert, and that black insert is shown about halfway up. It actually goes to the bottom of the cuvette, and with slits designed to allow the light sheet to enter. You'll measure the video at 9 for scattering at 9 degrees, and you have a little teeny stir bar that is very hard to see at the bottom of this photo, and that's what stirs the sample to bring up fresh particles. So that allows us to properly mix between measurements, fresh aliquot sizing, gives us nice statistics. This also means that we don't have any dead spots like in a flow system. We don't need to have any manual intervention. You press the go button, and then you can do something else for a few minutes while the instrument collects data. The removable insert means it's easy to take apart and clean. If you're working with highly concentrated protein samples, that's very important. And with other samples, generally it suppresses cross-contamination quite nicely. A typical lab will have multiple cells, and they'll make a sample and set up the next one, alternating cells or something like that. And because the expense of these cells is manageable. Okay, Sean's comment, everyone's favorite part, the math, and he is right, and it is all handled by the computer. Really, at the end of the day, you're using a Stokes-Einstein relationship to get from particle motion, which gets you particle diffusion, to get particle size of hydrodynamic diameter. We're going to track many particles. We're going to decide which of the 3 data sets. So as we use a color camera, and so you have a color video, which has a red channel, green channel, blue channel, one for each laser. And so the computer decides which set of lasers to use, or which laser color to use for each particle in a measurement. So we run it simultaneously, we have all this data, and then we do the processing afterwards. It requires a lot of CPU processing because you have a hefty data stream as a 3-color video or 3 video channels. And so we record and then process in the second session. This prevents us from dropping frames and missing crucial particles. And we do know that with higher speed cameras, that can be an issue. We see it in other techniques as well. So here is a quick video, and you'll see the 80 nanometer particle more or less in the middle, moving around quite rapidly. Then you'll see on the left side, the larger 700 nanometer particle, it's moving slowly. And then the 240 on the right, it's moving somewhere in speed between the 80 and 700. Now, of course, I talk about speed, but these are just kind of random motions due to Brownian diffusion. Jumping to another video to show some detail. So here I took that, well, it's a somewhat different sample, but I took that video you saw before, and I zoom in on one section. It would sort into a blue channel, a green channel, and a red channel. And then if a camera is saturated, like we did false color so that it shows as blue on the blue channel, green on the green channel, and red on the red channel. And so you see 3 sharp points in the upper left corner here, and you see that they're missing in the green and red channels. And so that tells you that these are weakly scattering particles. So if we didn't have the blue laser running, we would miss these small particles. If you look down here in the middle, you can see that I have large blobs of blue. Okay, and that large blob means that when I go to find the particle location, I'm going to do a pretty poor job. And the reason I do a poor job is because they're fairly asymmetric, and that's really due to scattering effects. There's nothing I can do about it in terms of optical design. And I've saturated the camera, which means that I can't do any sort of fitting around the shape of the peak or the intensity to find the center of the particle. Let's jump right to the red. Here we see the scattering. They're nice, sharp, small points, and it's very easy to find the particle locations. Well, really to get particle motion, you're really finding the particle in each frame. And so if you look at the far right red, we're going to get much better motion information compared to what we get from the blue. So for this size particle, we really want to work with the red laser signal because we're going to get useful information. Sometimes this gets much worse in the sense that these blobs will start to overlap each other. And then again, the lower power laser really helps you identify what's happening with your larger particles in a sample. So if you have a range of particle sizes, the take home is that you really can't pick one laser power and suit everything. You need to have multiple powers, which means multiple simultaneous lasers. So far I've talked about scattering, and scattering, you have light come in and the particle scatters light. It's coherent and it's the same wavelength as the incoming light. There is another optical phenomena that you'll stumble across, particularly, actually HORIBA has a rather large, very large spectroscopy divisions, and one of those is devoted to fluorescence. So if we excite the particle with some shorter wavelength, let's say in the blue, we'll see emission at some longer wavelength. That changes, known as the Stokes shift. So there are a couple of things about fluorescence. The first is incoherent, which really makes for a miserable dynamic light scattering experience, but does not bother this nanoparticle tracking because we don't rely on coherence. And the second is it lets us differentiate between particles that are fluorescent versus not fluorescent. And the way we do that is by using an optical filter. So I put in an optical filter, in the case of a long pass filter, shown in gray. Now I've blocked that incoming blue light on the left. So any scattered light that's the same as the excitation is blocked from the camera. And I only see the emission wavelengths in green. And so I pretty much made the non-fluorescent particles invisible to the detector. And that lets me identify a particular species in a sample. Here's an example of some results. This is a mixture of 3 carboxylate fluorescent beads, nominal 500 nanometers. We do have some background from the liquid. That's this extra peak you see at 200 nanometers. Now, if I were to block the incoming light, and I think we blocked the incoming blue, then you'll have this green fluorescence shown here. I'm sorry, we blocked the incoming green, we look at the signal. Here we block the incoming blue, and we look at the signal. And again, we can see some particles, and we don't see any of that dirt that was over 200 nanometer we saw earlier. And finally, we can play the game with red. So these are really 3 different types of beads, each with different fluorophores attached. And we're going to separate the 3 out. And so if I want to overlay the data, I have concentration on the left as a function of diameter. And you can see that we have the same peak at 500 nanometers that we expect. And we can recover that kind of by adding up the green and the blue and the red. And we don't observe any of the 200 nanometer impurities. And so with this fluorescence and appropriate tagging, you can really kind of separate out which species you have in your sample. I'm going to talk a little bit about some applications. Most of these tend to happen kind of by happenstance, I guess, in the eastern United States. Sean's been involved in it, and that's kind of why he was giving this talk and where we have the data. Working with a few private companies in Southern Maryland, Walter Reed Army Institute Research, and then the UMaryland Baltimore, the medical school with Niaz Khan. There are a few others. Someone reminded me, we forgot a couple, but in the area, but there you go. So I'm going to first talk about what is a lentivirus. So everybody's excited about coronaviruses these days, and we're looking at lentiviruses. Lenti is Latin for slow, and the best known virus in that family is certainly HIV and its various phenotypes, FIV, EIV, and so on. And lentivirus is interesting because it has the capability to introduce large amounts of viral RNA. So you can infect a host cell or even non-dividing cells, and you can create long-term stable cells with consistent gene expression. Yeah, so here's a quick summary of the full cycle from really transfection to transduction. Is you'll have what we'll call a packaging cell that generates the RNA, the proteins and so on to make up the virus. The virus buds off, comes out of that packaging cell. That's in a cell culture. And then we introduce it to some host cell where the viral RNA then forms DNA strands to be incorporated into the host cell. So now you can see that this is really a tool for introducing DNA into host cells that might be missing some critical piece of DNA. And that's really where the interest in lentivirus is. So kind of process, the virus binds to the exterior of the cell. We insert the RNA. The machinery cell creates the RNA-DNA complex, migrates to the nucleus where it's inserted into the DNA and causes the desired changes. Why lentivirus? Well, it's slower incubation time and creation of non-dividing cells means it reduces immune response associated with gene therapy. And so that's important, and you see where we're going with this comment on gene therapy. The goal is to introduce genes into a sick person that will make them better, if you will. So major hurdle in treating genetic disease is that our body has this well-tuned system for rejecting viruses. And so we need to find a delivery virus that won't overly excite the immune system. Now, Children's Hospital of Philadelphia has used a very similar process to cure congenital blindness in humans. It takes a lot of work, but you can make people see this way, and that's just huge. It's something you never would have imagined a few years back. So where does nanoparticle tracking analysis and multiple lasers fit in? Well, lentiviruses are about 100 nanometers. And so if we want to start figuring out what's going on, we're going to look at a technique like nanoparticle tracking analysis. They're efficacious as dimers and even as detritus with some RNA attached and aggregates and so on. So the goals were really, we want to measure the size of the virus in the aggregates, figure out what size particles we have in there. We want to get an accurate count across the size range because we want to figure out how many virus particles we have made in that initial transfection process. And could this virus count correspond to infectious titers? Well, let's back up and say, what is infectious titer? It's really a measure of how well a virus is able to infect target cells as a measure of concentration. So I have X infections over Y amount of virus provided. So if you figure out how much virus do you need to get the cell effect you want. Well, there are a few methodologies to measuring infectious titer. The viral plaque assay is time tested. It's kind of a gold standard of virology. It's cheap, it's effective, it's time consuming, but that's good. On the other hand, it's time consuming and it depends on the users for accuracy. So by the time you throw in skilled users and time consuming, then you find out that people want to get paid. It doesn't look so cheap anymore. A second is quantitative polymerase chain reaction, qPCR. It is accurate, it's expensive, and it does require virus-specific primers. And so you kind of have to have your whole system very well characterized to use qPCR. Viral plaque assays, you serially dilute your virus, and then you infect plates of confluent cells. That's shown in the figure on the right. And then you incubate until you start seeing cells dying. Now, if you have a slow-moving virus like lentivirus, this process could take a while. And so you might be spending 2 weeks waiting until you get a reliable number of cell morphology changes and deaths. when you've waited long enough, then you apply the agar gel just to slow diffusion and kind of freeze everything. And you count the number of cytopathic incidents for each of the dilutions. And you average to get your infectious titer. All right, that all sounds great, except there's some user-to-user variability. The cells don't necessarily wave a little cross and say, hey, I'm dead. So people have to judge whether a cell has changed enough to be called infected or dead. And so that's an issue, because you and I might disagree on what is a weird-looking or dead-looking cell. And then it does take time. Someone's sitting there staring at a microscope. So the goal with the ViewSizer is really find a range of sizes over which we can collect data that gives comparable reproducible number that correlates to the infectious titer assays. We measure the whole spectrum of sizes for each sample, and then we looked at the results really based on kind of three windows. Look at one virus, just a very narrow size range, the virus and its dimer, so that's a somewhat wider size range, and everything, detritus up to virus aggregates. And so I don't want to show real data here, but the results really broke itself. This proposal is we have three ranges. In the black, I show, this is concentration as a function of diameter. And the black, we show, say, monomer or single virus units, dimer in the blue, and then the green, which is kind of everything. The distribution in the background is a polystyrene latex sample, which just shows the resolution of the nanoparticle tracking analysis technique for a well-defined sample, which, of course, a virus titer is not. So viruses tend to have a broad range of sizes in the sample. The data collection, we analyzed across multiple replicates for each retained sample and across multiple users, including Sean and employees of the company that we were working with. Compiled everything to get an average and standard deviation for each sample run, applied sample count for each size range versus known infectious titer values, and the results indicated that measurement over a wide range of sizes gave the best predictor of infectious titer value. So you want your best R-squared values in the 0.9s, you really need to measure a wide range of particle sizes. Yeah, so that range up to make up a number 400, and infectious titer, we're typically in the order magnitude apart, but we need to see a range of sizes to get accurate results. And one thing about this virus production is, in spite of the cartoons, the viruses produced are not perfect. You get some virus wall pieces, you get some aggregates, you get some monomer, some dimer, and you really need to look at everything to correlate well with actual virus activity. So some takeaways, they did have a single laser system, and the application didn't work for that. With the multi-laser system, they were able to find a consistent and robust correlation. It cut their time frame for figuring out what was happening in their system from several weeks to around 20 minutes per sample. And just having that data so quickly helps you understand your manufacturing, lets you get much tighter reliability and reproducibility. It was crucial to measure the entire range of particles in a sample and correlate it to infectious titer. And you can look in literature to see similar work with viral vectors. So there's citations here. Let's compare to other techniques. qPCR requires prior knowledge of the viral genome sequence, because you have to derive primers against non-mutating regions of the genome. And you have to make standards that you'd really derive back from these plaque assays, which is the same thing that we're doing with ViewSizer and looking at concentration. It really, qPCR just looks at small specific regions of nucleic acid. And so you're not looking at the entire package. Is the nucleic acid embedded in something that will get itself inside of a host cell? You don't have to lyse cells for nanoparticle tracking analysis. Dilute and run. And so that's helpful. And then the accuracy for qPCR, of course, depends on purity, because you could have side reactions. So nanoparticle tracking is fast and is going to be important in the viral therapy market. And it's reproducible across multiple end users. So your only work to do is serial dilution from the original aliquot and then press the measure button. Now, that was really the big kind of headline that came out of that effort, and they're very happy about that. But there's some other things that you can do once you have this instrumentation. You can look at stability testing if you do freeze-thaw cycles. Now that you have some inexpensive measurements with the nanoparticle tracking and the multi-laser system, you can say, hey, let's freeze-thaw this as I go in and out of -80 freezer and see what happens to my material. And that's going to give me some important information about product volatility. It's also, of course, important for looking at batch-to-batch variability. So you can use a QC tool if you don't want too many aggregates, or if you have other cell detritus, you can see it with the multi-laser nanoparticle tracking analysis. And you can look at production and either predict or better understand process upsets. Okay, shifting gears from viruses, which kind of unusual or should be unusual in your body, to something that's very common, which are extracellular vesicles. And so these are vesicles that are released by many different cell types, both during your normal life as well as when you're pathological states. Extracellular vesicles are found in all sorts of biofluids. You'll find it in blood, you'll find it in bile, and it gives you a potential for long-range communication, brain to gut, brain to peripheral immune system. Because they're in your blood, you can obtain novel biomarkers, and because they affect physiology, you can almost think of using them for therapeutics. So let's just kind of zoom in on one area. If you have central nervous system injury, then one working model is, hey, you've injured these nerve cells, you're going to have changes in your circulating extracellular vesicles, and that can cause systemic dysfunction because of the signals they're sending, particularly if the signals are too high or persist too long. So if you have a working model, you can say, okay, does this model make sense? And then you'll go back and ask yourself this question: are there changes in number and size distribution profile of plasma extracellular vesicles after spinal cord injury? So if the model is right, then you certainly would expect something like a yes answer to this model. Experimental design is you start with, I think it was mice, you do a spinal cord injury or no injury, you collect blood, you obtain the plasma without platelets, and then you can separate further to large microvesicles or small microvesicles and extracellular vesicles. And you can analyze the size, distribution, and concentration of each by nanoparticle tracking analysis. So this was some work that they did comparing a single wavelength system versus a single wavelength of the ViewSizer and against all three wavelengths. And the size distribution is shown in the upper left, and the black points are the single laser system. The gray crosses are what you get from all lasers. You notice how the size distribution has shifted to the left, and so we're getting smaller particles in there. And with the red only, you see very much like what you got with the single laser system, where you have the peak and distribution about the same place and a tail off to these larger size particles, which certainly exist in the sample. Yeah, so a couple comments. One is there's a lot going on that you're missing if you just try to analyze with a single laser. And because you could tune your single laser to look at 200 nanometers, and then you'll lose kind of the smaller particles. So a significant portion of the sample is really needs all three wavelengths. And you can better understand this polydispersed sample using a multi-laser system like the ViewSizer. So here is another set of results with the three lasers on particle detection. The black points on the size distribution kind of shown in the middle is all three. Blue only, you can see the peak of the smaller sizes. Green is the peak kind of peaking up here at the middle sizes, and the red only is the largest sizes and the lowest concentration. A couple lessons here. You can certainly see from the overall concentration that you need all three lasers, and you can also see that the red and the green are a lesser part of the sample than the smaller particles. And so you really need this higher energy laser to get accurate analysis of the smaller extracellular vesicles, and you need the lower power laser of the red and the green to get the larger ones. Moving on to something, I guess this would be the opposite of where I started this, which is adjuvants. So as you go to make a vaccine, which you want to use to suppress a virus, you might add adjuvants as particles to excite the immune system and at the same time not make the healthy patient sick, if you will. So you put in something kind of neutral that the immune system recognizes as, hey, I got to respond to this, but doesn't actually go and reproduce and wreak havoc. And so this is the Army liposomal formulation. This is some work at the Walter Reed Army Institute of Advanced Research. And this was recently published in Biochemical and Biophysical Research Communications. And you can see a fairly broad size range coming out of this adjuvant, where you have a peak at a small particle size, and then you have a very significant number of larger size particles in the adjuvant. And then the solid dashed lines reflect the sample as made and then after lyophilization and reconstitution. And of course, you can see where that's going, which is they're interested in lyophilizing the vaccine and then reconstitute before injection as a way to preserve it. But you can use data like this to reassure yourself or tell yourself what kind of changes you can expect in the sample as you bring it through different processing steps. Another thing that turned out to be important is that you had to track not just this narrow peak at the smallest particle sizes, but you also had to look at the larger particle size adjuvant particles. They certainly are there, and they certainly seem important physiologically. And just the fact they're there means you want to keep score in case they change and they do things you don't want them to do. So kind of conclusion, NTA is a powerful tool for looking at polydispersed samples that are difficult to impossible to properly characterize for size and concentration with other tools. Electron microscopy just takes too long to do a statistically significant number of particles, for example. For biologics, looking at size ranges from about 50 nanometers to 2 microns, that's kind of the size range that works well with this technique. You can expand it somewhat with fluorescence, and you can also use fluorescence to interrogate subsections of complex mixtures. And we've made significant progress in areas of viruses, gene therapy, extracellular vesicles, exosomes, and adjuvants, as well as several other areas. And so I'll kind of close with a thank you. I'm not going to read this to you, but when this goes online, you can see these citations and for further reading. Now I think I'm going to open the floor for questions.
Thank you, Jeff. I know it's not easy to present a different deck of slides organized by Dr. Sean Travers, but I think you did a great job. For those of you who have
Those
questions,
are great slides.
They are really good slides.
That's really what helped. That's all I gotta say. They were great slides.
In fact, I think it's very worthy of applications notes, and that is to say that we have an applications note on our website for virus, exosomes, proteins specifically. If you have any questions, you can definitely send it over to labinfo@horiba.com. Dr. Travers will be able to help us out. So, question: can the ViewSizer analyzer be used to detect EVs labeled with fluorochrome conjugated antibodies in a similar way that flow cytometry does for antibody label cells?
Yes. Yeah. So, and in fact, Sean has some promising preliminary work demonstrating exactly that point. Unfortunately, kind of the HORIBA labs in New Jersey got shut down for a while, and so we haven't kind of put the nails in that coffin yet. But the short answer is yes, and the somewhat longer answer you need is we have some idea how to go about it. So there you go.
Yeah, it's definitely a tricky time to maneuver. The other question is, can using a fluorescence tag that is efficient, can it possibly look at smaller biologicals?
That's another area that we're actively working on now. And again, it's things just kind of got bollocksed up with lab shutdowns. We don't have access to a BSL-2 facility with live viruses, which is where we want to chase that. But yeah, we have some strategies for that, and that needs some testing before we can say exactly which ones work the best.
Perfect, thank you. Now, this question came specifically from the registration. Can it meaning the ViewSizer 3000 nanoparticle tracking analysis, can it measure small to intermediate peptide to peptide non-covalent complexes?
No. And so, all right, yeah. Let me give the longer answer because I think it's based on a lot of assumptions that about the question. So I'm thinking when you say small to intermediate, you're thinking monomer, dimer, trimer type peptide aggregates, if that's the right word. And I know from experience that when we work with proteins, even full proteins, that neat proteins that are well behaved in solution, we do not see anything. So if you take a lysozyme monomer or BSA monomer, they just don't scatter well enough to see the individual protein molecules. And that's the same to hold for the peptides. Monomer dimer, again, they don't scatter well enough. When you start getting up to larger size aggregates, if you get up to 50, 80 nanometers, that's where we can start doing the counting and sizing analysis for these aggregates. So when you say medium size, I'm assuming you mean under 40 nanometers, and that's why I led with a no. That said, DLS will work well in that size range, but does not give you really useful concentration information.
Thank you for the thorough answer. Now, the next question is, how long does it take to run a single sample?
So it takes about 15 minutes. And so, yeah, I think it's 12 to 13 minutes. I throw in 2 minutes for kind of bobbling the cell or whatever stupid things I do at that time. So anyway, that's where the 15 comes from. Now, one thing you can do to boost throughput is you could run a bunch of samples at 6 minutes each, and then do all the analyses overnight when you're not in the lab at 7 minutes each. And so you could, if you really need to boost throughput, you could do it that way.
Thank you. Yeah, I think we know labs who run the instrument on a 24/7 basis. Now, the next question is, could you label smaller proteins?
Yes, we can. Again, you're going to have to, particularly if you're labeling a protein, you're not going to get that many labels on it. There is some potential to use gold nanoparticles as labels, and then that would work.
Thank you. What about the concentration of volumes, volume necessary?
Okay, so the necessary volume is about 450 microliters. The concentration is somewhere from about 1 e to the 7 particles per ml to 1 e to the 8 particles per ml. Yeah, incidentally, the concentration range is a bit wider than that, so I'll just tell you kind of where you're going to have the most fun. But you can run lower than 1 e to 7. You can run up to about 2 e to the 8 particles per ml. If you're in DLS, here I'll show my biases, you're looking at about 100 to 1,000 times more dilute than a DLS sample.
Okay, thank you. The next question is it possible to measure particle autofluorescence and distinguish what subpopulations of particles have that property?
Okay, so I'm trying to figure out, so I assume by autofluorescent you mean that the particle incorporates fluorophores on it, and so you can distinguish distinguish particles with fluorophores from particles without fluorophores. And so I don't know if that answers the question exactly, but that's probably the best I can say right now.
Thank you. Are there any solution conditions that are incompatible?
Yes. So, okay, so if you have an aqueous solution and it contains a strongly absorbing molecule in it, then you could find that, so if you have molecules, I think vitamin B does this to me. It absorbs strongly. I think it absorbs the blue or the green. I forget which. And so you might be forced to turn down the laser power because of that. That doesn't happen a whole lot. A more common request we get is with an organic liquid. And if you have an organic liquid that likes to heat up quickly and likes to absorb, and I'm thinking of halogenated compounds because they seem to like ammonia solutions, which, okay, I'm sorry, that's another aqueous system, but carbon tetrachloride, you really can't do much for data, but you can use things like alcohol. And then if you have simple salts that aren't doing anything optically, knock yourself out. The other thing is we're going to measure every particle in your system. So if you bring me a diluent that is loaded to the eyeballs with particles, I'm going to teach you how to clean it up, and then we'll run your sample. Yeah, so we need clean, but I can help with that. We've been through that a number of times.
Thank you. So going back to the person who asked the question, can the data be processed in a separate computer?
Yes, yes. So people who really push it through for throughput can purchase a second license to the software and then collect data on one machine and process on a separate machine. So yes, that can be done.
Thank you. Can the system be used to size submicron oxide particles?
Submicron oxide? Oh yeah, they scatter very well. In fact, yeah, we can go below that. I mentioned that 50 nanometer number earlier, but if you start looking at things like oxides, we get down to smaller sizes.
Right, definitely. I know when we talk about NTA, a lot of times we focus our applications on life sciences, but this is definitely an instrument that's very compatible with oxides as well.
Yeah, I mean, it's been used with metals, it's been used with oxides, it's been used with coatings, which are basically polymeric nanoparticles. So
cool. Thank you, Jeff. Okay, so I got one more question that came in. Is that okay, Jeff?
Yeah.
Can the analysis detect dynamic interactions between particles?
Okay, so I'm going to answer that question a number of different. It's a really interesting question. I'm glad I waited. So there are a couple different things when we talk about particle dynamics. The first would be, are we going to use the instrument to look at changes in your system over time and get at questions of particle kinetics? Say, are we having nucleation growth phenomena? Are we having, how are my particles changing? And because we can extract both concentration and size, we can really get at those questions much more readily than, say, light scattering, where you're looking at the scattering over time, which is kind of a combination of concentration and size effects. The other is if you want to look at interactions between particles, say long-range electrostatic interactions. We don't have anything in the software that does it, but we can output the particle tracks. So that's the good news, is if you have a way of thinking about it, you can get some, it looks like you can get some interesting data. The bad news is that the concentration is extremely low. The particle concentration is extremely low, and that means that kind of your traditional long-range interactions that get into, say, charge interactions between latex particles is pretty negligible because the particles are so far apart. And the reason we go to low concentrations is so the particles are distinct when we're trying to do tracking, because we don't want the particles to cross, because then we never figure out who comes out the other side. So if we can find a system with a very long-range interactions, there's some games you might be able to play with the tracks. And then if you have a system that you want to track things like nucleation phenomena, this is a great tool and kind of a big step above current approaches, which just rely on total scattering. So good question. Thanks. I hope I helped.
Yeah, it did. Thank you so much. And I think that's all we have for today. Is there any concluding comment that you'd like to add before we say bye to our audience?
No, I don't know if Sean's still online, but I want to thank him publicly for the slides.
I am here. Can you guys hear me?
I can. Yeah. Anyway, thanks for all that.
I just wanted to thank Jeff for doing this, and I didn't want to cough/wheeze my way through this. I've been under the weather recently, so thank you, Jeff. And if you have any questions individually, if you send them to us, I'd be happy to answer them as best I can.
Great. Thank you, everyone.
Thank you all. Okay.
Alrighty.
Thanks.
Bye.
Bye.